So the folding process that goes on inside a cell with every polypeptide that's produced, the folding process is dependent upon the sequence of the amino acids. It's the chemistry of the R groups on each amino acid that will allow for hydrogen bonding to occur, or van der Waals interactions to occur, or other types of weak attractions to help this polypeptide fold up. The molecular chaperone proteins are going to bind to polypeptides so that the folding is carefully monitored and only allowed to occur in one particular way, producing that one three-dimensional shape that that particular protein needs to be in. The charge of the R groups, the polarity or non-polarity of the R groups, all of that matters. Hydrogen bonds and van der Waals interactions are going to hold those R groups together in the ultimate folded version of the protein and allow it to do its job correctly. Now, while many proteins get folded up into the correct shape on the first try, there are some proteins that will be misfolded. The good news is that many misfolds are corrected while they're happening. If suddenly in the midst of folding, a hydrogen bond is taking place where it shouldn't be taking place, heat shock proteins or chaperone proteins can release from the polypeptide and then reattach and correct that problem. But sometimes the mistakes that are made during the folding process are not caught right away. And the cell ends up going through and folding up a protein incorrectly. Folding up of proteins is so important to a living cell that there are sort of proofreading systems in place to deal with any protein that is misfolded. I have two of them listed here. The first one is referred to as the unfolded protein response, or the UPR, and the second one is called ER-associated degradation, or what we call ERAD. These are two processes that are in place in living cells to deal with the problem of a misfolded protein. So abnormal folding matters because any protein that is not folded correctly is going to be a protein that can't do the job it was intended to do. Remember, form and function are intimately related when it comes to proteins and other molecules in the cell. Let's think about the unfolded protein response first. When a protein gets folded up inside the rough endoplasmic reticulum lumen, that protein is going to interact with the wall of that lumen, or the membrane of the rough endoplasmic reticulum. Remember, if the protein is sitting inside the lumen of the RER, it can bump into and interact with the inner monolayer of the RER membrane. There are sensors in that inner monolayer that can detect a misfolded protein. Something else that I want to state here. Remember that HSP70, or binding protein, BIP, sits right near the translocon inside the RER lumen. When that polypeptide is passing through the pore of the translocon, as it's being translated, as it's being made, BIP can bind to it before that process is finished. In other words, BIP can start interacting with a polypeptide while it's still coming in to the lumen of the RER before the translation process is finished. Remember, we're talking about hundreds to thousands of amino acids in a polypeptide. That means that many individual molecules of BIP are going to have to bind to that polypeptide in order to guide the folding process. And those BIP molecules are going to start binding as soon as they can, as soon as they're exposed to the polypeptide coming in to the lumen of the RER. So when the sensors in the inner monolayer of the ER membrane, when they detect that folding has started and there's been a misfold somewhere, one of the first things that's going to happen is translation of that polypeptide is going to stop. All right? It's just going to stop momentarily. We have a problem, in other words. The cell is saying, we have a problem. These first few folds that are being made are wrong. Now, you might ask yourself, well, why doesn't the BIP just let go and rebind and fix that fold? Well, part of the reason it happens is because we may need additional BIP proteins at this moment to do that important work. So when the misfold is detected and translation is temporarily halted, that allows the cell to increase the translation of more chaperone proteins, and even some proteins that are involved in degrading misfolded proteins. Those proteins are referred to as proteasomes. Misfolded proteins are going to be refolded, or I should say unfolded, and then refolded, using a lot of these newly made chaperone proteins. This just goes to show you how much a cell is willing to invest in the folding process for proteins. This goes to show you just how critical it is that proteins are folded correctly in living cells. Living cells that are constantly translating brand new proteins. This is going on constantly in cells. Living cells are willing to constantly adjust how much chaperone protein is available so that those chaperone proteins can, on the fly, adjust how a protein is folding up. Any misfold matters in a protein. It has to be detected, it has to be addressed, it has to be fixed. The unfolded protein response is all about that. It's all about detecting the misfolds, unfolding where the misfold is, and then refolding to get it right. Now, this can happen all the way throughout the folding process. But there will come a point, as I said a moment ago, that a protein is finished folding. And sometimes proteins get to that point with misfolds in them. In other words, sometimes the unfolded protein response fails. Proteins get misfolded sometimes. When that happens, the ERAD system is there to fix the problem. Now, what the ERAD system is going to do is not unfold and refold the protein, but go ahead and destroy this misfolded protein. The ERAD system is capable of recognizing a misfolded protein, retranslocating it, in other words, moving it back out of the lumen of the RER, and sending it into the cytoplasm so it can be degraded. Folding of proteins is so important to a living cell that if a protein manages to get through the folding process and turns out to be folded incorrectly, if a protein ends up folded incorrectly, the cell will destroy it rather than have it floating around in the cell. Proteins are powerful things, and it's much better for a cell to just destroy this thing that it has just spent a lot of time and energy making. It's much better to destroy it than allow it to go out there and potentially do damage. Why do we care? Why does a cell care so much about folding? The answer is that proteins are powerful things. A misfolded protein is not going to be able to do the job it was intended to do, but that doesn't mean it's incapable of doing any job. Sometimes misfolded proteins in a cell will go on to do things that they were never intended to do, and that can lead to the disease and the death of the organism. The most commonly referred to example of this is the prion. And that's what you're looking at on this slide in the drawing. You may have heard of prions, or what are sometimes referred to as prions. Prions are misfolded proteins that cause terrible neurodegenerative diseases. One of the most commonly studied neurodegenerative disease is actually a disease in cattle called mad cow disease. We have had in recent decades sporadic outbreaks of mad cow disease. Not so much here in the United States, but in other parts of the world. In the drawing on the left-hand side, you're looking at just a normal protein that circulates in the bodies of healthy cows and has a normal job to do. You can see that the drawing shows the protein in its folded shape. The areas in green are where there are alpha helices. The area over here in blue is where there are beta pleated sheets. And then you can see these yellow sort of strings connecting the helices and the sheets. Those are just strands of amino acids that don't get folded. They don't attach to other areas. So this is what this protein should look like in its folded shape. Sometimes, though, this protein folds up like this. And this is what we call a prion. So this protein, as you can see, has one fewer alpha helix and many more beta pleated sheets. It's in a different shape. This misfolded version, this prion, is not going to be capable of doing the job that this protein was intended to do. Unfortunately, it is capable of causing some damage. If you're familiar at all with mad cow disease, what makes it so damaging and devastating is that these prions will gather in nervous tissue and they will accumulate with one another and damage the neurons themselves, causing them to die. And that process leaves literal holes in the brains of these affected animals. These are referred to as spongiform diseases because the brain, like I said, literally contains holes in it the way a sponge contains holes. There are a couple of particularly frightening things about prions. One is that once a prion or prions are present in the brain, they can cause normally folded proteins to misfold. In other words, they can create more prions. So it's almost as though a prion can amplify its numbers by causing other proteins to misfold. Another really scary feature of prions is that they are transmissible from one species to another. So human beings who consume the nervous tissue, including the brain tissue, of a cow that has mad cow disease, can then go on to develop that disease in themselves. And there are versions of these terrible spongiform diseases in almost all animal species. Very, very frightening example of how a misfolded protein can go on to devastate the health of an organism. On this slide, the artist has put together a lot of what we've been talking about so far in terms of cotranslational import. Now, in the drawing, you can see the endomembrane system that we mentioned earlier in the lecture. Remember, the endomembrane system includes the nucleus, the rough endoplasmic reticulum, transport vesicles, the Golgi apparatus, and the plasma membrane. All of this membrane is involved in sorting proteins in the cell. The artist has blown up a little section of the rough endoplasmic reticulum membrane in order to show us cotranslational import in action. Remember, any polypeptide is going to be produced on a ribosome near the rough endoplasmic reticulum, a ribosome that's here in the cytoplasm near the RER. If that polypeptide is intended for its final destination to be one of the organelles on our list, and it's intended to go through cotranslational import, then the very first amino acids that are going to be translated on this polypeptide are going to be an ER signal sequence. The signal recognition particle is going to bind to those 15 or 20 amino acids, and then the signal recognition particle is going to bind to its receptor in the translocon. The ribosome will be brought down and it will bind to its receptor on the translocon. The signal sequence will come into the pore and bind to the wall. And then as the ribosome continues to translate the mRNA, the growing polypeptide will pass through the pore into the lumen of the rough endoplasmic reticulum, as we talked about earlier. Chaperone proteins are going to start to bind to this polypeptide as soon as it starts coming in, so that the folding process can begin and misfolds can be prevented. Eventually, the entire polypeptide will be translated. The signal peptidase will cleave the signal sequence off. The ribosome and the mRNA will disperse. The polypeptide, which has now undergone some folding, will be released into the lumen. Remember, we said this polypeptide is going to travel through the endoplasmic reticulum. Other modifications will be made. Chemicals might be added on, small molecules like methyl groups and acetyl groups. We might also see bits and pieces of it cleaved or spliced. It may get glycosylated. It may have some carbohydrate added to it. And as it reaches the end of the rough endoplasmic reticulum, it's going to be placed into a transport vesicle and sent along to the Golgi apparatus, where additional modifications will be made, particularly to the carbohydrate side chains that have been added to the protein, if there are any. It will then be packaged into more transport vesicles and sent off to its final destination. Any polypeptide, any protein that's produced through this system of co-translational import is going to go to one of the destinations that was on our list. Rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, lysosome, plasma membrane. Those are, and cytoplasm. Those are the destinations of proteins that are included in co-translational import. Now, the last thing to talk about in this lecture today is the other process that I mentioned at the very beginning of the lecture, what's called post-translational import. There are some proteins that aren't destined for any of those structures on our list. For example, there are proteins that are destined to remain floating in the cytoplasm. There are proteins that are destined to become part of the nucleus, or the mitochondria, or the chloroplast, or the peroxisome. Any protein whose final destination is in this list is not going to go through co-translational import. Instead, it's going to go through what we call post-translational import into these final destinations. These proteins, as they're being translated on ribosomes, are going to be translated on free ribosomes that are floating in the cytoplasm, not ribosomes that are going to come down and bind to the translocon. These proteins are going to be translated on these free ribosomes. They may or may not get modified. They will be folded eventually, but they will be brought in to their final destination through a process known as post-translational import. After translation is complete, after this polypeptide has been made, it will be delivered to its destination and it will be imported into any of these structures so that it can perform its final function. Now, we're not going to talk about any of the particular steps of post-translational import. All I need you to know is that these are the organelles where post-translational import occurs. These are the organelles that those proteins have as their final destination.